हेलो गाइस, आज मैं आया हूँ अपने नए वीडियो के साथ जिसमें हम लोग एक बहुत ही इम्पोर्टेंट टॉपिक के बारे में डिस्कस करेंगे। पहले हम लोग ने देखा था कि फ्लावर क्या चीज़ होता है प्रीवियस चैप्टर्स में, कि फ्लावर में आपका मेल पार्ट होता है और आपका फीमेल पार्ट होता है, और वही मेल पार्ट और फीमेल पार्ट का डबल फर्टिलाइजेशन होके पोस्ट फर्टिलाइजेशन चीज़ें होते हैं, और उसमें से एक चीज़ आपका होता है जिसके बारे में आज हम लोग डिस्कस करेंगे, ये 4 लेटर वर्ड सीड। सो लेट्स मूव ऑन टू दिस टॉपिक। पहले हम लोग थोड़ा सा रिकैप कर लेते हैं प्लांट का। अगर आपने प्लांट चैप्टर अच्छे से पढ़ा होगा, मतलब एकदम इन डेप्थ स्टडी की होगी, तो आपको पता होगा कि ये क्या है। यू कैन इजीली आइडेंटिफाई दिस एज द फीमेल पार्ट ऑफ द फ्लावर। So what is this? This is the stigma. Everyone knows that. Style or the pollen tube by which the pollen grains from other plants or other flowers or the same flower, the anther of the same flower, the pollen grains shall travel, and then it goes inside. It goes further inside to and it reaches the part where it has its final destination. You can say it reaches the ovary. So this is the path it takes. It goes into the ovary, then there is double fertilization, and then finally the flower modifies into a fruit. But what does this part fertilize to, change to? It changes to a seed. And then after that, after forming into a seed, when we, like for example, when we consume an apple, we are eating that apple, but we don't consume the seeds. Why? Because there is actually no matter in it, so that we can consume it. So we just throw it, and if we throw it or if we plant it in a garden and with proper conditions, if it gets the proper conditions, then it will germinate. Just concentrate on the word that I'm using. It will germinate to form another apple tree gradually. So this is like the most basic part I have done, and before moving on, you should know this. If you don't, please don't watch this video, because I will be going into I will be showing you a deep study of seed. So if you don't, I'm just cautioning you, if you don't know what I have done, don't watch the video. So as you can see, I have drawn the inner part of the ovary of a flower. Okay, so this is the ovary, and starting like this is my tube, the pollen grains travel and it goes through this opening, which is called the micropyle. Okay, and this is a very important part in the seed when it forms into a seed. Okay, now it goes into the egg cell. This is the egg cell of the flower. It goes into the egg cell. Now these are my two polar nuclei, that are my pollen grains. Okay, nuclei. Okay, and then one of this will fertilize with the first set of egg cell, and the other polar nuclei will fertilize with the other set of egg cell. And so we have something called as a double fertilization. I'm not going into the deep of all of this because I am assuming that you know all of this. Okay, so then it forms into a seed. Now when it forms into a seed, it has a specific structure, and so if the flower is a monocot flower, that means the seed has I'm just writing it, monocot or a dicot. Monocot, cot full form is cotyledon, and if it is a dicot flower, then if it is a dicot flower, this will happen. If it is a monocot flower, there will only be single fertilization. There will not be double fertilization. Okay. Come on. As you can see, I have made diagrams of a bean seed. Actually, it's the same bean seed. So a bean seed looks something like this. A bean seed looks something like this. If you see, one sec. If you see this white part, can you see this white part? I think it's can you see this white part? Yeah. So this white part is my micropyle. Okay, and if I zoom a bit, there. Okay, wait. The below the white part is my hypocotyle, and above is my epicotyle. And this, okay, see, you can see that hole. Can you see that hole? Yes, that is my okay, wait one sec. Can you see that hole? Sorry. Okay, so this hole is my hypocotyle, and above the white part is my micropyle. So my so actually the white part has been covered. Yeah, the white part has been actually, it is, if you see, it is the roots, I mean the baby roots that are coming out. So in our seed, the bean seed, that is actually called rajma. So you can go in the kitchen and just look out for rajma, and if you soak it in water and then after one day you check it, you will see some roots structures coming out, some structures coming out of the seed. That will be my soaking for 2 days. Okay, soak it for 2 days. You will see some roots like structures or some structures coming out. That is your radicle or the baby root, that will form into a big root when it is soaked into soil. So the white part that you saw in the seed was actually the radicle, or that will form into a root. But if I kind of magnify it, I will, and if I just remove the white part, I will see a small hole that is called the micropyle. Okay, it is the same hole, same opening that allowed the pollen grain to enter the egg cell. Okay, sorry for this arrow. This arrow will only extend to the dot. This is called the epicotyle, and this is called the hypocotyle. I'll explain everything. Okay, this is called the tegmen because it is going inside. This is called the testa. This is called the cotyledon. Okay, so this is like a basic, just I've done it for the sake of it. Epicotyle grows into the plumule, and that plumule grows into the shoot. Hypocotyle grows into the radicle, and then root. The micropyle, it just comes out when the seed fully germinates, and yeah, and cotyledon is basically this part. Testa is also called the seed coat, and this seed coat comes out from the seed when it germinates, and this seed coat is a very high source of nutrients for the soil. Okay, and tegmen is basically like the cell membrane. It just protects the protection. Okay, it just protects the cotyledon. Okay, this was like the one part. When you split it up after um, soaking it for 2 days or more, then you get this kind of structure. Don't break it. If you break the cot, the two cots, if you break it, then it will not work. You have to split it up in such a manner that it doesn't, the two cotyledon don't separate each other. Okay, now this is nothing but my epicotyle, and this below is my hypocotyle. I'll explain everything, just wait. Testa or the seed coat, tegmen protects the cot. This is one cot, cot 1, and this is the other cot, and this thing over here is my micropyle. This one is my rudimentary leaves. Okay. So I'll explain everything. Now, what is the epicotyle? Epicotyle is what just happened? Okay, epicotyle is nothing but it's just a kind of structure that grows into the plumule and then the shoot. It is just like a baby shoot in a bean seed or in a seed, and hypocotyle is just the opposite. It is the baby root of a seed. And yeah, so now we have, I think everybody knows what is germination. Germination is the process of it's like simply a process when a seed transforms into a plant. That's it. Okay. Now for germination to happen, there are many conditions that that the seed should be provided with. First, warmth, that it gets under the soil. Second, oxygen. Third, water. Fourth, what's the fourth? Optimum temperature. Okay, these are the conditions that the seed should be provided with: warmth, oxygen, water, and optimum temperature. I think this is all clear. And if any of this thing, if it is not given, like if optimum temperature is not given, the seed will not transform into a plant, a proper plant. Germination is classified into two types: hypogeel germination and hypogeel and epigeel germination. And what happens in epigeel? It's like a simple concept. It's a very simple concept, and you should not lose your marks in this concept. So, if this is my surface and my seed is over here, in epigeel germination, the hypocotyle, it grows faster, it elongates faster. So what happens is, it elongates faster. That means first it develops its roots, and then it pushes the plant upwards, and then the epicotyle also simultaneously but slower elongates. Okay, now this and with this the plant is taller, and this mostly happens in dicot leaves. I'm sorry. This mostly happens in epigene germination. It mostly happens in dicot leaves. Sorry. Okay, and in hypogene germination, the epigene epicotyle elongates. So if this is my seed, first it just some roots, because the hypocotyle is slower in elongating, and then some leaves, because the epicotyle is faster in elongation. But when the epicotyle is elongating faster, the plant will be not so tall, because the epicotyle has no support, because the hypocotyle has not elongated that fast, and so the foundation or the roots are not that strong, so for the epicotyle to sustain. So so hence the plant is not so tall, and in this the plant is tall, it is short. Okay, these are some basic differentiation between epigene germination and hypogene germination. Okay, so epigene is just the opposite, like epi, you have to just replace the word with hypo when it is comes in part of the elongation, and you have to just reverse it. Don't make any mistake in this. Okay. As you can see over here, I have drawn a maize grain. That is also an important chapter. So we were talking about bean seed, that was a dicot seed, and now we'll move on to maize grain, that is a monocot seed. Okay, so this is also an important seed. So we'll just mark out everything. So this thing is obviously the testa, and this thing, we'll not mark this, we'll mark this afterwards. This is the endosperm that protects the cotyledon, and what is this? Okay, we'll mark this afterwards. This is the scutellum, or simple words, the cotyledon, and okay, we'll mark this afterwards. And this is the plumule, radicle, and all of this is my embryo. Okay, now the things which I have not marked are very important, but I'm not marked. 1, 2, 3, and 4. These 4 things are like the most important parts of a maize grain. What is this in your books? It is given. Let me check if my spelling is right. Yeah, it is right. Alluron layer. Then we have the epithelium. And over here we have conjunctile. Yeah, and then we have the coleorhiza. Okay, now these terms are quite confusing. I know that. Even I took a lot of time to learn these, but it is not that tough. So this testa over here has a special name in a maize grain, that is the fused pericarp. Don't make it pericarp, it's pericarp. And this pericarp is like the fusion of the endocarp and the mesocarp. The mesocarp and this thing. What is the outer layer called? Come on, epicarp. Yeah, epicarp and the mesocarp. It is just the fused. pericarp. Agrelon layer is a special layer that protects the maize grain, and endosperm is like the matter that is present above the epithelium. Okay, epithelium is the separating part between the endosperm and the scutellum or the cotyledon. Scutellum is nothing but the cotyledon. Coleoptile, okay, plumule. What is the plumule? It is, okay, it forms into a shoot, radicle, it forms into the root. Coleorhiza and coleoptile are two main things that we have to learn in this maize grain. Coleoptile, coleoptile is a part of the plumule that is like the upper part of the plumule, and what was I saying? Yeah. So it actually protects the plumule, and the coleorhiza protects the radicle. But it has a special feature of apart from just protecting it, it helps the plumule to form into a shoot, and the coleorhiza helps the radicle to form into a root. But this only happens in a maize grain. Now, a maize grain follows hypogeel germination. That means the epicotyle elongates faster. Okay. Now, the epicotyle elongates faster. So, epicotyle over here is nothing but the coleoptile. Just remember that. So, the coleoptile is elongating faster. That means if this is my maize grain, it looks something like this. No, it looks something like this. So, the maize grain, this will form this, the shoot will be the rate of the formation of shoot will be faster, and the roots will be not so fast. That means the roots will not be deep. That means you can make a conclusion that this is weak and this is quite strong. Okay. So yeah, that's it for today. Now I wrap up the chapter seed by concluding that this chapter is not difficult, but it is not easy either. You have to just focus on the epi, hypo, and coleoptile, and just if you just break the word like epicotyle, if you just break the word epi, just means above, cotyle, what can it be? It can just be supportive or just like that. Even I have to figure that out. But if you just memorize it, you will just forget it after the test or your exam. But if you concentrate on the concept, how it is done or how it is formed, and logically, practically, if you think, only then you can master this chapter. Thank you.